Good morning, good afternoon, good evening, class. You are welcome to today's lecture. In today's lecture video, we will be discussing about chemistry of amino acids and proteins, lecture 2. In the previous lecture, we discussed about amino acid, which we said they are the monomeric units or the building blocks of proteins. And there are 20 common amino acids that are found in proteins. So before we understand protein, we need to know that these amino acids are the building blocks, and we have already highlighted that in the previous lectures. So for we to know the protein constituent, we need to determine the amino acid composition of that particular protein, and also we need to go ahead and carry out the determination of sequence, that is the sequence of the amino acids that are found in the protein. So in this lecture, we will be discussing about how to determine the amino acid composition of proteins, and how to determine the amino acid sequence of a particular protein. We cannot discuss about sequencing without giving credit to the first person that tried to work on insulin. The name of the person is Frederick Sanger in 1953. He was actually working on a protein, and he was able to isolate and sequence the protein insulin, which is a hormone, a peptide hormone, insulin that is important in carbohydrate metabolism. So he was the first person to work on that protein and determine the amino acid sequence of that protein. And he was given a Nobel Prize in chemistry for that wonderful work in 1958. So let's look at how to carry out purification of proteins. So if you want to purify a protein, you need to determine the amino acid composition of that particular protein. So for you to purify it, for you to determine the chemical composition and structure and function of that particular protein, that protein must be purified. So the first task is to develop an assay that is to see the procedure that you can use for the purification of that protein. So the procedures, depending on which type of protein you are working on, and where is the location of that protein in the cell. Is it inside an organelle? Is it an organelle protein? Is it found within the surface of the cell membrane or inside the cell membrane? Okay, so you need to have an idea of where the location of that protein and the specific property of that particular protein. So, for you to develop that assay, you need to know that each protein has its specific property, and such properties, such as the rate of transformation of substrate to products, which is if the protein is an enzyme, we need to talk about the rate of transformation of substrate to product, and antigen, antibody, antigen reaction, or a physiological response that give a quantitative measure of the activity per unit protein concentration. So if you are talking about the protein activity per unit, okay, per unit protein concentration, you are talking about the specific protein or the specific enzyme activity. So the purpose of a purification procedure is to increase a protein's specific activity to the value expected for the pure protein. So in initial purification of a soluble cellular protein involves destruction of the cell membranes. So if you are talking about a protein that you want to isolate from its soluble cellular components, so you need to destroy the cell membrane. It means the protein you are targeting is inside the organelle rather, or maybe inside the cellular components. So you are not talking about, it's not a membrane protein. So you need to disrupt okay, destruction of the cell membrane. Then you carry out centrifugation, okay, differential centrifugation in a density gradient to isolate the protein from subcellular particles, and you carry out high molecular weight aggregates. So essentially what you are doing here is after you disrupt the cell membrane, you now carry out density differential centrifugation, because each of the organelles, they have their own density. So you carry out centrifugation at various speed, different speed, depending on what you are targeting there. It's actually a procedure that you can follow to isolate that particular organelle, so as now to continue with your purification for demo. So if you want to carry out that, you will further carry out purification procedures which may utilize selective precipitation by inorganic salt, which we call salting out, by using organic solvent. And there is what this final purification steps, which include combination of techniques that can separate the protein based on either molecular size or the molecular charge of the protein. We talk about the affinity of that protein towards maybe a substrate, okay, if it's an enzyme or if it's an antibody or antigen that you want to do. So you talk about the affinity of that protein towards that substrate. So you do affinity chromatography, or you can do combination of this, two or more of these techniques, so that at the end of the day you have your purified protein. So there are techniques based on charge, which include electrophoresis. We have isoelectric focusing and ion exchange chromatography. So these are techniques that you separate or purify your protein. These techniques, they are based on charge of that particular protein. So there are those techniques that are based on molecular mass, which they include molecular mass or size, which include ultracentrifugation, molecular exclusion chromatography, or size exclusion chromatography. You can call it gel filtration. And for affinity chromatography, it is based on the affinity of that protein for its substrate. You can talk about membrane receptors or antibodies if it's an antigen. So there is also what we call polyacrylamide gel electrophoresis, which is based on both size and charge. So how do you determine the amino acid composition of a peptide or protein? So the first thing to do is you have your peptide or protein, you hydrolyze it into its constituent amino acid. You know, the protein is made up of amino acid. So when you have that protein sample, now let's say this is your protein sample in a test tube, or the peptide, okay, in a test tube, you know, the constituent individual building blocks are the amino acids. So you need to hydrolyze it into what? The component amino acid by heating with 6 normal hydrogen chloride. Okay, you use 6 normal hydrogen chloride at 110 degrees Celsius for 24 hours. So this will now break down the peptide bonds and hydrolyzes now the protein, so that you get your constituent amino acid. So you have now, after that, you have your amino acids that are found in the hydrolyzed. Now, since you have taken your time to do that digestion, at the end of the 24 hours, you should know that inside your test tube is no more that peptide intact or protein intact. It is rather the amino acid that are found in the what? Hydrolyzed. The hydrolyzed is your sample, your HCl that you use, okay, and the solution that you are working with. So then you are going to separate this hydrolyzed by ion exchange chromatography on a column of sulfonated polystyrene. So you use sulfonated polystyrene as your column in the chromatography setup. So the technique you are going to use is you use what? Ion exchange chromatography, which is when this one is separating the the component based on what? Based on charge, on the charge, individual charge of the what? Amino acids. So you now have the separated amino acids. You can now detect them by color produced when they are heated with hydrogen. So now you have your amino acids differently. You don't know what are they. You don't know which amino acid are there. So you now put forward for another reagent, which is ninhydrin, and this ninhydrin will now react with the alpha amino acid to give an intense blue color. Okay, so if there is an amino acid in the solution, when you react with ninhydrin, it gives you an intense blue color. So you should know that when you see blue color, it means you have your what? Alpha amino acid. But this test is not specific for amino acid. It can also pick up other compounds that have amino groups. Okay, so this is like a general test for compound that have amino groups, because amino acid have amino groups, right? So it will give like a general idea that, okay, there is amino acid, but which amino acid you don't know which and which, right? So, and if the amino acids like proline are present in the solution, you will see a yellow color. So it will give you a yellow color. So now the quantity of the amino acid is also proportional to the optical absorbance of the solution after heating with hydrogen. So how much of those amino acids are present is proportional, is a function of the word absorbance that you will get after you must have taken your time to measure the absorbance in a spectrophotometer. So there is also a fluorescamine, which is used which is used to react with the alpha amino acid group to form a highly fluorescent product. So this fluorescamine on its own is not fluorescent, but when it reacts with the alpha amino group of the amino acid, it forms a fluorescent products. Okay, it forms a fluorescent product that can be quantified using fluorimeter. Okay, spectrophotometry using fluorimeter. So the identity of the amino acid is revealed by its elution volume. So what is this elution volume? It is the volume of the buffer that you use to remove the amino acid from the column. You remember when you hydrolyze, let's go back a little bit. When you hydrolyze with cis normal hydrogen chloride, you allow it at 110 degrees Celsius for 24 hours. Then that digest that you have after 24 hours, you run an you run a chromatography using ion exchange, right? Ion exchange chromatography. But the column that you use, it was a sulfonated polystyrene column. So now, what is this elution volume? Now, for you to know the identity of the amino acid, is we say you are going to use now buffer to like rinse the column. You are putting the buffer into the column so that it will now bring those amino acids that are inside the column. So you are now collecting in a tube. You are collecting each of your eluate now in a column. So the chromatographic pattern of the hydrolyzed set is then compared with that of a standard mixture of amino acid. So when you say standard mixture of amino acid, this is a mixture that contains a non-concentration of the amino acid. So you use that pattern that you get from your own digestion to compare with the standard so that you will know, okay, this is for this amino acid, this is for another amino acid. So when you determine the amino acids composition, now how do you determine the sequence of the amino acids? How do you know that which amino acid is linked to which amino acid at what position? Because the first step that you did, you just determined the amino acid. You don't know, you just determine the composition of the amino acid, but you don't know, is it alanine that is together with, that's bonded with glycine before the tyrosine? You understand in the sequence. So now you now determine the sequence, this one after the other. This amino acid now bonded to form a peptide bond with this amino acid, then form a peptide bond with another amino acid. That's sequence. So sequencing of protein, it was first carried out by Frederick Sanger in 1953. He determined the amino acid sequence of insulin. So matured insulin consists of 21 residues in chain A and 30 residue in chain B, and they are linked by what? Disulfide bonds. So Frederick Sanger reduced he reduced the disulfide bonds using performic acid, and thus he separated the two chains. So he separated the two chains using performic acid. He then cleaved each of the chain into smaller peptide using this reagent. He used trypsin, chymotrypsin, and pepsin. These are the reagents that he uses to further break down each of the peptide in chain A and chain B separately. So the resulting peptides were then precipitated them with 6 normal hydrogen chloride to hydrolyze the peptide bonds and generate peptide with as few as 2 or 3 amino acids. So after that, he reacted each of the peptide with a reagent that it was named after him. Sanger's reagent is 1-fluoro-2,4-dinitrobenzene. So this is the Sanger's reagent, which derivatizes and the exposed amino group of the amino acid. So it uses this reagent now reacts with the what amino group of the amino acid. Okay, so working backwards to larger fragment enables Sanger to determine the complete sequence of insulin. So he used that technique to now determine the complete sequence of the insulin. So there is also what we call densyl chloride, which is used to identify the amino terminal residue. So when we say amino terminal residue, you remember in the previous lecture, we said these are the amino acids that are at the terminal end of the, or at the terminal end of the sequence. Is it N or when you have your sequence of the amino acid, you have the amino terminal, right? You have the amino terminal and you have the carboxy terminal, right? So the amino portion is the first sequence used. The amino acid that is the amino terminal residue is the first amino acid in the sequence. So you use densyl chloride to identify which of the amino acids at the first residue in the sequence. So it will react with the amino groups to form highly fluorescent and stable sulfonamide derivatives. So although this the DNP and densyl methods the Sanger's reagent and densyl method are used, they are powerful. They cannot be used repetitively on the same peptide because the peptide is totally degraded in the acid hydrolysis step. So you cannot use both the Sanger method and densyl chloride because you have this degradation of the peptide. So another scientist that is called Brad Edman introduces another reagent, which is phenyl isothiocyanate, which is called Edman's reagent, which selectively labels the amino terminal residue of the peptide. So it generates PTH, which is phenyl thiahydrantoin, which is a derivative in contrast to the Sanger's reagent. So the phenyl thiahydrantoin derivative can be removed under mild condition to generate a new amino acid amino terminal residue. So a successive round of this derivatization with Edman's reagent can therefore be used to sequence many residues of a single sample of peptide. So Edman's sequencing has been automated using a thin film or solid matrix to immobilize the peptide and use HPLC to identify PTH amino acids in the solution. So HPLC is high performance liquid chromatography that is also being incorporated in the procedure. So the Edman method determines the first 20 to 30 residues of a peptide. So if you have a peptide that is very long, so when you use Edman method, it determines the first 20 to 30 residues. Then, since most of the polypeptides contain several hundred amino acids, it is necessary to first cleave the polypeptide into small peptides prior to what? Edman sequencing. So if you want to use Edman sequencing, you have to hydrolyze the polypeptide into smaller peptides because it is limited to what? The first 20 to 30 residues of the peptide, so that you have smaller peptides which you can use the Edman reagent to carry out the sequencing. So the agent for the chemical or enzymatic cleavage of protein include cyanogen bromide, you have trypsin, etc., etc. So we are going to see in the subsequent slide where the reagents and the cleavage site in this table. Okay, these are the reagents used in the cleavage of the polypeptide, and these are the reagents, and this is the cleavage site. So for chemical cleavage, we have cyanogen bromide, which it cleaves at carboxyl side of methionine residues. So it's at the carboxyl side of methionine residues. This is very important. Uh, you need to understand that cyanogen bromide is a chemical reagent that is used in the sequencing of amino acid. So you have cyanogen bromide, which it cleaves the carboxyl side of methionine residue. So wherever there is methionine residue, it will go to the carboxyl side and cleave. So you have 2-nitro-5-thiocyannobenzoate. So it cleaves at the amino side of cysteine residue. So wherever there is cysteine residue in the polypeptide, it will go and cleave on the amino acid side. It will go now, amino terminal side, it will now go and cleave it. So you have these two are what chemical reagent, and we have the enzymatic reagent, which are trypsin, clostridium, staphylococcal protease, and hydroxylamine. So these are what enzymatics. Now trypsin cleaves at the carboxyl side of lysine and arginine, while clostridium cleaves at carboxyl side of arginine residues, while staphylococcus protease cleave at the carboxyl side of aspartic acid and glutamic acid, while hydroxylamine cleaves at the asparagine glycine bond. So if there is asparagine glycine bond, it will now cleave that bond. So let's look at, we are still on the purification of proteins, but for us to understand it, we need to know what are the levels of protein structure. There are 4 levels of protein structure. We have the primary structure, secondary structure, tertiary structure, and the quaternary structure. So primary structure refers to the sequence of the amino acid and the location of the sulfide bridges if there are any. So it is a complete description of the covalent connection of the protein. So this is just like a sequence of the amino acid. We have alanine, glycine, lysine, and so on and so forth. So that is just it. If there is a sulfide bridge, it will show that. So it's just a complete description of what the covalent connection between the amino acid. So this is just a simple sequence. It's called the primary structure. So what about the secondary structure? The secondary structure is in the next level after primary structure. Refers to the steric relationship of amino acid residues that are close to one another in a linear sequence. So some of these relationships are of irregular kind, giving rise to a periodic structure. We have the alpha helix and the beta pleated sheath, and also the collagen helix. These are the 3 examples of what secondary structure. So the helix is a rod-like structure. The tightly coiled polypeptide main chain forms the inner part of the rod, and the side chains extend outward in a helical array. The stability of the alpha helix arises primarily from hydrogen bonds formed between the amino group and the carboxyl groups of the main chain. The carboxyl group of each amino acid is hydrogen bonded to amino group of the amino acid. So that is situated 4 residues ahead in the linear sequence. So for you to know, I will show you the structure of the primary structure, the secondary structure, and as well as the tertiary and the quaternary structure, so that you see the picture of how they look like. So for the rotation, we talk about the alpha helix. There is a rotation which gives 100-degree rotation, which gives about 3.6 amino acid residue per ton. I will show you. So let's look at this. This is alpha helix. Look at it. This is alpha helix. That is turning. So this turn from this is 3.6 amino acid residues. So, and this is the carboxyl terminals, and this is the what amino terminals. So the carboxyl terminals, you have the carboxyl functional group, and the amino terminal is what the amino functional group that is there. So you have the beta plated sheet. You remember alpha helix and beta plated sheet, they are what, and collagen helix, they are all the examples of what secondary structure. So the beta plated sheet, it forms when two or more polypeptides line up side by side. So when you have two or more polypeptides, they line up side by side, they form what we call the beta plated sheet. While for the alpha helix, you have this kind of structure. So each individual polypeptide in the beta strand will have this to be this is the beta strand, okay? So we have individual polypeptide that line up side by side. So each of the beta strand is fully extended, and you have stability, being stabilized by hydrogen bond between the amino group and the carboxyl group of adjacent chains. So you have two types of beta plated sheet. You have the parallel, which you have the amino terminal and carboxyl terminal of each of the polypeptides. They are facing towards the same direction. So you have amino, if this is peptide A and this is peptide B, so their amino terminal are lined up adjacent to each other, and their carboxyl terminal are also lined up. So they are facing the same direction. While antiparallel, if this is the amino terminal of this polypeptide, this is going to be the carboxyl terminal of second polypeptide. So for collagen fiber, this for collagen helix is found in collagen protein, and this is how it looks like. So what about tertiary structure? Tertiary structure refers to the entire three-dimensional conformation of the protein. So it's the 3D structure, 3D conformation of the protein. It is the steric relationship of amino acid residues that are far apart in a linear sequence. It includes geometric relationship between distant segments of the primary structure and the positional relationship of the chains with one another. It indicates in a three-dimensional space how secondary structural features, that is the helixes and the sheet, assembled to form what we call the domain. So for the quaternary structure, this refers to the arrangement of the polypeptide chain in a multi-chain protein. So if you have a protein that is multi-chain, it has many chains. There are proteins that have one chain. There are those that have two chains. Some have four chains. We will see example. So the arrangement of this polypeptide in the chain, that will give you the quaternary structure. So the subunits in a quaternary structure are associated non-covalently. It should be noted that protein having only one polypeptide chain, like myoglobin, as well and as one which the polypeptide chains are covalently bonded together. So e.g. chymotrypsin cannot have what we call quaternary structure. Okay, so it means proteins that have only one chain, like myoglobin and chymotrypsin, they don't have what we call quaternary structure. Okay, so because they just have one chain, but those that have two chains and four and more chains, they can have what quaternary structure. So each polypeptide in a protein with quaternary structure is referred to as a subunit. For example, hemoglobin has 4 polypeptide chains, 2 alpha chains and 2 beta chains. They are held together non-covalently in a specific formation as required for its function. So a protein domain, now a protein domain is referred to the compact semi-independent globular unit of a protein structure. That is what we call the domain. It is a section of protein structure capable of performing a particular chemical or physical task. So for tertiary and quaternary structures, they are stabilized by a non-covalent interaction, which include hydrophobic interaction, hydrogen bonds, soil bridge, and intra-bond polypeptide disulfide bonds. So these are the types, the representation of various types of the protein structure. This is the primary structure, which is the arrangement of the amino acid in sequence. You have the carboxyl terminal, and this is the amino terminal. In the secondary structure, you have the alpha helix, and you have the what? Beta plated sheet. Alpha helix, and what? You have the beta plated sheet. And you have tertiary structure, but the three-dimensional structure, and the quaternary structure, you have multi-chain. Okay, you have this is a different chain, this and that chain, this and another chain, this is another chain. So when they come together, they form what we call the quaternary structure of that particular protein. So what are the roles of proteins in biological process? They play a vital, important role in biological process, and this include enzymatic catalysis. You remember that enzymes are proteins in nature, so many of the biological activities that is taking place, many of the chemical reaction in the biological system is being catalyzed by enzymes. So they play enzymatic catalysis because these enzymes are what? Protein. So it's also another function, another role is the transport and storage. So many small molecules and ions are transported by specific proteins, e.g. Hemoglobin. Hemoglobin transports oxygen in erythrocytes, while myoglobin transports oxygen in muscles. So you remember there is this disorder of blood, which is the sickle cell anemia. You remember this hemoglobin is a protein that is found in the red blood cell. So it is important to study these proteins and their structure and their composition, because this, at the work of Frederick Sanger, enables scientists to now detect or trace evolutionary changes in protein structure, and also to detect evolutionary relationship between the changes that occur in the protein and some diseases at molecular level. So if you look at sickle cell anemia, if you look at the hemoglobin that has 4 chain, beta chain, 2 beta, and 2 alpha. In one of the beta chain, there is a mutation that is changing of the amino acid. Instead of glutamic acid, now the amino acid, glutamic acid, is being replaced with valine. So in sickle cell disease, you know, because hemoglobin responsible for the transportation of oxygen in the erythrocyte, so that sickle blood or hemoglobin that is having that mutation cannot carry out that function. That is why you have a lot of crises in people that are having sickle cell anemia, just because of that mutation from one glutamic acid to valine. Okay, so and also that we have transferrin, which carries out iron in the plasma of blood to liver, where it stores it as a complex with ferritin, which is another protein. So you see the function of transport and storage. We have what we call coordinated motion proteins that are the major component of muscles. Muscle contraction is accomplished by sliding motion of 2 kinds of protein filaments. On the microscopic scale, coordinated motion such as the movement of chromosomes in mitosis and the propulsion of sperm by the flagella also produced by contractile assemblies consisting of proteins. So they also help in coordinated motion. There's also mechanical support that exceed high tensile strength of skin and bone is due to the presence of collagen, and which is a fibrous protein that is found in the skin. So we have immune protection. Antibodies are highly specific proteins that organize and combine with protein substances such as viruses, bacteria, and cells from other organisms. So we have also under function generation and transmission of nerve impulse. The response of nerve cells to specific stimuli is mediated by receptor proteins, e.g. rhodopsin, which is the photoreceptor protein in the retinal rod cells. So we also have function of this protein in control and growth of growth and differentiation. There is controlled sequential expression of genetic information, which is essential for the orderly growth and differentiation of cells. there's what we call repressor proteins, which are important control elements that silence specific segment of DNA of a cell. Okay, these repressor proteins, they are control elements. They silence, okay, now the DNA, which is having the information for the production of a particular protein, is going to be silenced so that that protein can be produced because it's not needed. Okay, and because of that, the expression of that gene is very essential and is time and energy consuming, so that is why it needs to be water regulated. So repressor proteins are important that control by silencing the segment of that gene so that the protein cannot be produced. We have nerve growth factor, which is a protein complex. It serves to guide the formation of neural networks in higher organisms. So these are some of the functions or roles of proteins that are found in the biological system. So thank you very much. This brings us to the end of today's lecture. Please endeavor to watch the video and also read your course material and ask questions in the forum where you have issues. Thank you. See you in the next